Hello students, let us start with the next chapter of 11th standard. This is a 14th chapter, that is respiration in plants. So what are we going to study in this chapter? In this chapter we will study about respiration in plants. We will study about glycolysis, fermentation, aerobic respiration, the respiratory balance sheet, Glycolytic pathway, and the respiratory quotient. So first of all, we need to understand the difference between respiration and breathing. Breathing is only exchange of gases, whereas respiration is oxidation of food materials to get energy in form of ATP. So this energy is used for different purposes like absorption, transport, movement, reproduction, and breathing. We all know that photosynthesis is the ultimate source of food, right? We get all the food from photosynthesis which occurs in the chloroplasts. So as we all know, during photosynthesis, carbon dioxide and water are used. With the help of light energy, glucose is made in chloroplast. So compounds that are oxidized during respiration are called respiratory substrate. So here, carbohydrate, which is mostly glucose, is oxidized to get energy. This is cellular respiration. So generally, the carbohydrates are the ones which is oxidized. They are the most common substrates. The energy released is not used directly, but it is used to synthesize ATP. ATP is adenosine triphosphate. Hence, ATP acts as the energy currency of the cell. So in plants, oxygen and CO2 are taken in and given out. This happens with the help of stomata. Stomata is the small opening which are surrounded by the guard cells and lenticels. One difference that we notice, plants do not have any specialized respiratory organs like humans. We humans, for example, have lungs. So why is it that they don't need any specialized respiratory organs? This is because each plant part takes care of its own gaseous exchange need. So gas transport is very limited. Gas exchange is very low as compared to animals, and the leaves are adapted for maximum exchange during photosynthesis. So during this, oxygen is released within the cell. So what actually happens during respiration is glucose is oxidized and we have formation of carbon dioxide and water, and lots of energy is released in form of ATP. This energy is used to synthesize other molecules. During the glucose catabolism, if you remember, is breaking down of substances. Not all the liberated energy goes out as heat. Glucose is oxidized in several small steps, which we are going to study in detail, and these steps enable it to release energy in form of ATP. So during respiration, oxygen is used and CO2, water, and energy are ultimately released. Certain organisms are adapted to anaerobic conditions, so they can respire anaerobically in absence of oxygen. So some are facultative anaerobes and others are obligate. Now let us study the glycolysis pathway. Glycolysis is basically the breakdown of glucose to pyruvate, right? So here there is partial oxidation of glucose to two molecules of pyruvic acid in absence of oxygen. So this is also called as EMP pathway based on the name of its founders, and it occurs in cytoplasm of all living cells. So this scheme was given by these scientists Gustav Amden, Otto Meyerhoff, J. Parnas. That is why it is called the EMP pathway. In anaerobes, it is the only process in respiration. So this is where respiration ends for anaerobe with the synthesis of pyruvate and glycolysis pathway. So what happens during glycolysis? So we know that glucose is derived from sucrose or from the storage carbohydrates, right? In plants, the storage carbohydrate is starch. So this sucrose is converted into glucose and fructose with the help of an enzyme called invertase. Now these two monosaccharides, they enter the glycolytic pathway. So what happens in glycolytic pathway is the glucose and fructose are converted into glucose 6-phosphate. Here phosphorylation is taking place. Enzyme hexokinase is involved. And then this glucose 6-phosphate is converted into fructose 6-phosphate, right? After that, the steps for metabolism of glucose and fructose. So the ATP is used in two steps. First, in conversion of glucose to glucose 6-phosphate, and then in conversion of fructose 6-phosphate to fructose 1,6-phosphate. These are all 6-carbon compounds. So what happens after this is fructose 1,6-bisphosphate is split into two compounds, that is dihydroxyacetone phosphate and phosphoglycerol aldehyde. These are both 3-carbon compounds. The phosphoglycerol aldehyde is oxidized with inorganic phosphate and it gets converted into bisphosphoglycerate, right? So during this, two redox equivalent are removed from phosphoglycerol aldehyde and they are transferred to NAD plus and we have NADH and H plus. So this bisphosphoglycerate then becomes 3-phosphoglyceric acid, which is also called as PGA. So this actually yields energy. As we can see, ATP is made here, right? So when this ATP is formed, energy is trapped in this process. ATP is also formed when PEP, that is phosphoenol pyruvate, converts to pyruvic acid. In glycolysis, if we observe, 4 ATP molecules are directly synthesized from one glucose molecule. Pyruvic acid is the key product of glycolysis. It is a 3-carbon compound. So it can have different metabolic fates depending on the cell's need. So this is the entire glycolysis pathway. We start from glucose and we end up with pyruvic acid, which is a 3-carbon compound, and 4 ATP molecules, 2 here and 2 here, will be made during this process. So this pyruvate has 3 different fates. It can either enter lactic acid fermentation, alcoholic fermentation, or aerobic respiration. These two reactions occur in absence of oxygen, whereas Krebs cycle is in presence of oxygen. So fermentation is incomplete oxidation of glucose under anaerobic condition. It occurs mainly in prokaryotes and unicellular eukaryotes. So there can be two types of fermentation, alcoholic or lactic acid. Here, the pyruvic acid formed from glucose is converted to ethanol, right? And carbon dioxide is released. So the enzymes pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. This occurs in organisms like yeast, right? Net ATP production here is 2. Okay, so total only 2 ATPs will be formed from one glucose molecule. So yeast poisons themselves to death when the concentration of alcohol becomes too high. When it becomes 13%, then it will not be able to survive. Then we have lactic acid fermentation. Here, pyruvic acid is converted into lactic acid. This occurs in some bacteria. Here, net production of ATP from one glucose molecule is 2. So in animals, when oxygen is inadequate during exercise, pyruvic acid in muscle cell is reduced to lactic acid by the enzyme lactate dehydrogenase. So whenever we exercise, we often see that we have cramps in our muscles. So that is because of lactic acid formation in our muscle. Here, the reducing agent is reoxidized to NAD plus in both type of fermentations. So there are some drawbacks of these fermentation. First of all, the energy produced is very limited. Less than 7% of the energy in glucose is released and not all of it is trapped in form of ATP, and hazardous products are made. These are acids and alcohol. So let's see what happens in aerobic respiration. Aerobic respiration is the complete oxidation of organic substances in presence of oxygen, and it gives carbon dioxide, water and ATP. It occurs in mitochondria, that's why it's called the powerhouse of the cell. So for this, pyruvate, that is the final product of glycolysis, is transported from the cytoplasm into the mitochondria. So there are two crucial events in aerobic respiration. First is the complete oxidation of pyruvate by stepwise removal of all the hydrogen atoms, and it leaves 3 carbon dioxide molecule. This takes place in the matrix of the mitochondria. The second important step is passing on of electrons which are removed as a part of hydrogen atoms to molecular oxygen with simultaneous synthesis of ATP. This occurs in inner membrane of the mitochondria. So the pyruvate enters the mitochondrial matrix and we have oxidative decarboxylation. This occurs in presence of an enzyme called pyruvic dehydrogenase. It requires coenzyme NAD plus and coenzyme A. So during this process, two NADH molecules are produced from two pyruvic acids. The acetyl-CoA then enters the Tca cycle. So ultimately we have pyruvic acid which is converted to acetyl-CoA. Now this Tca cycle is also called as Krebs cycle after its founder. It was first elucidated by Hans Krebs. It is also called as citric acid cycle. Let us see what happens in this cycle. So these are the steps of the Tca cycle. It starts with condensation of the acetyl group with oxaloacetic acid and water to form citric acid. In presence of citrate synthase enzyme, a coenzyme molecule is released. This citrate is then isomerized to isocitrate, and then decarboxylation of isocitrate leads to the formation of alpha ketoglutarate. So this now is a 5-carbon compound. Decarboxylation of alpha ketoglutarate acid to is converted into succinyl coenzyme, succinyl CoA. So the next step in the cycle is that succinyl CoA is converted into succinic acid. This is called substrate level phosphorylation. A GTP molecule is also made here. So in a coupled reaction, GTP is converted into GTP with simultaneous synthesis of ATP from ADP, triphosphate from diphosphate. Then this succinic acid or succinate is oxidized to fumarate, and then it is converted to malic acid or malate, which is again a 4-carbon compound. Then we'll have oxidation of malate. So for oxidation, the NAD will be converted into NADH and H+, and then we'll have oxaloacetic acid. At 3 points of the Krebs cycle, NAD plus is reduced to NADH and H plus. We can see here these are the 3 points where these happens. At 1 point, FAD plus is reduced to FADH2. So the continued oxidation of acetyl-CoA by Tca cycle requires continued replenishment of oxaloacetic acid. It also requires regeneration of NAD and FAD from NADH and FADH2. If we look at the entire cycle, then we have pyruvic acid, which is converted to 3 carbon dioxide molecules in the mitochondrial matrix, right? So here NAD is converted into NADH, water, ADP and phosphate are involved, and in turn we have 4H plus, FADH2 and ATP. So a glucose is broken down to give 6 carbon dioxide, 8 NADH plus H plus, 2 FADH2 and 2 ATPs.